All right, welcome back to part 6 of DNA replication. This time around, we are going to start looking into eukaryotic um, DNA replication, and we'll be able to compare and contrast the origins of replications in bacteria and eukaryotes. We'll also outline some of the key different polymerases that eukaryotes use. They use multiple, just like bacteria do. We'll also discuss how RNA primers are removed in eukaryotes, because this is slightly different. So, to start off with, um, eukaryotic DNA replication is not quite as well understood as bacterial. The more complex organism you get, the more difficult it is to piece apart the different elements involved. But we do have a lot there, right? So there are similar proteins in eukaryotes that we've seen in bacterial conditions. So, the fact that the same enzymes exist in both locations suggest similar processes. When we do genetic knockouts, we end up with similar results. Now, eukaryotic DNA does have a bit more going on, such as having linear chromosomes. That's an issue that we didn't have to deal with in bacteria, um, the process of replicating the ends. They also tend to be much larger, and consequently, we need to process that DNA replication much more quickly. The DNA is all packed up around nucleosomes, so we have to have a mechanism for moving through those. Um, and also, cell cycle regulation is just simply more complicated in eukaryotes. All right, one of the first major differences is that as opposed to a single origin of replication in bacteria, we have multiple origins of replication in eukaryotes. And this is necessary because the DNA is so much longer. Um, DNA replicates bidirectionally from each one of these origins of replications, and sooner or later they meet up with one another. Now, the origin itself, found in eukaryotes, has some similarities to bacteria, particularly when you're looking at lower organisms. So when you look at yeast, you've got these, what we call ARS elements, or autonomous replicating sequence. They're about 50 base pairs in length. They have a high percentage of A's and T's. Remember, this bonds more weakly than G's and C's. So if you can bind a piece of DNA, wrap it around a protein complex, if you have an AT-rich region, you can cause a separation to occur. And again, if you look at these sequences, they have some similarity in a general sense to the sequences we see in bacteria. When you start getting more and more complex in terms of eukaryotic organisms, it's a little bit less defined. And it may well be that the proteins that are inducing the beginning of this process are cueing in on chromatin structure, not sequence. And this is generally true in a lot of areas, that the chromatin structure itself is dictating how things progress. Now, replication begins with the assembly of what we call the replication complex. This is a slew of many different proteins that are coming together, but they are doing things such as recognizing, at least in yeast, they're recognizing particular sequences. But they act in a similar way, causing the DNA to bind and wrap around and bubbles to open up. Once that is loaded on, that will bring in the origin of replication complex, um, which will continue the process. So up here, what we have is largely chromatin remodeling occurring as a precursor to the rest of it. Ultimately, though, we do bring in a helicase, and this is where things start becoming more and more similar to the bacterial part of it. So once we have assembled all these things on there, get the helicase on, then the next thing to load on would be the polymerase. And like bacteria, eukaryotes have different polymerases for different function, but we take it to a whole new level. So, before we were talking about, 5 different polymerases. Now we're dealing with well over a dozen. And the first thing I want you to recognize is that the vast majority of these different polymerases have a specific function in DNA repair, or in specifically replication of damaged DNA. We have a polymerase that is specific to the replication of mitochondrial DNA. We have polymerases that are involved in the initiation process, and these are acting in conjunction with primase. And then we actually have different polymerases, one acting on the leading strand and one acting on the lagging strand. So let's start off, and again, you don't need to memorize all of these. I do want you to recognize that different polymerases have different specific functions. This concept of these first 3, I would key in on that, so that there is one polymerase that initiates and lays down the RNA, for the primase lays down the RNA, so that's an RNA polymerase, but it is working in direct contact in conjunction with the polymerase that starts adding in. But also that we then switch and are going to be utilizing these two, which is one on the leading strand, one on the lagging strand. All right, so the alpha polymerase is the only one that is associated with primase. So it is responsible for first primase laying down the RNA primer, and then the polymerase starts adding on DNA to that, to only about 20 to 30 nucleotides in length. And this will just do this job over and over again. Once it's gotten a little bit past this point, it switches and exchanges the alpha for the other two. So here again, you'll see that biologists love to use Greek characters to describe different elements or different subunits or different classes or types of things. So, for those of you that aren't familiar with Greek characters, this right here is the alpha, this is epsilon, and this is delta. So it's we tend to use those as opposed to others. So in any event, we have the alpha, which is in association with primase. The primase lays down about 10 RNA nucleotides. the alpha lays down 20 to 30 DNA nucleotides, and then we switch, right? And we refer to this as being the polymerase switch. And we swap out depending on which strand we're talking about. So the epsilon is involved in the elongation of the leading strand, where delta is used for the lagging strand. Now, we also have a slight difference in on the strands once we reach um, where we start to overlap with a primer. In bacteria, if you recall, there is 5' to 3' exonuclease activity that will come in and just digest away um, these different RNA primers. With eukaryotic organisms, we have something called a flap endonuclease that comes in and starts chewing it up. So the general concept here is instead of the polymerase itself having the endonuclease activity, what the polymerase actually does is start peeling up the RNA primer and just laying the DNA underneath it. And the free exposed 5' end that's hanging out here is subject to the endonuclease activity. So, a little flap will come up, it'll start getting digested away, and we'll continue through that process until we've made it all the way through.